Welcome back! You had a few topics without me, but I hope you haven't missed me too much, because now we're going to talk about protozoa. These organisms are members of the Protista kingdom, depending on who you ask. Some people don't have them in their own kingdom anymore, because some Protista are more similar to animal cells and are named as protozoa, which are the ones we're going to talk about, and some are more similar to plant cells. So they're up for discussion on classification. But right now, we're just going to focus on the ones that are like animals, and these are the protozoa. So why is this important? This introduces one of the three eukaryotic organisms we're going to be talking about this term. So it's important to know the basics of the protozoa. Here are our objectives for this topic. As always, I expect you to have these mastered before you move on to the next topic, and don't be afraid to ask me any questions about them. I'm more than happy to sit down and explain every single one of them if you need to. So what are protozoa? Their name comes from the Greek meaning first animals, and as I said, they have a lot of functions similar to animals. They're single-celled and they can live in water and soil, and most of them are harmless, but there are some parasitic protozoa. There's about 65,000 species of them, so there's quite a wide array of them, and some of these are the ones that we saw when we were doing the pond water in lab. So now let's talk about the protozoa and function. And I know there's a lot on this slide, so just try to hold on. Once again, this is trying to serve as a little bit of an outline for you so you can go back to this later and just get the big high points of them. They're always single-celled, and they contain almost all of the main eukaryotic organelles, but they don't have chloroplasts. Their cytoplasms are divided into two parts, the ectoplasm and the endoplasm. The ectoplasm is on the outside layer, and it's clear, and it helps provide for locomotion, feeding, and protection. So this is the parts that participate in phagocytosis and crawling. Whereas the endoplasm is more like that traditional cytoplasm, where it has it contains organelles and has all the jelly-like substance that we're talking about, and it houses the nutrients and other parts. Overall, it's really important to remember we just said there's 65,000 species, so there's a lot of diversity within these cells. So we got the euglena we just saw. This is an amoeba on this cell, so there's a lot of difference between these two. So they also, while there's a lot of difference, there's some really big similarities. Protozoa are known as heterotrophs, which means that they have to acquire their food. We already talked about how they're more similar to animals, so they have to acquire their food from various sites. And this is in comparison to those autotrophs, which are the plants, and the protozoa, or the protists, that are more similar to plants. Where do the protozoa get their food from? They scavenge them from dead plants or animal debris. They graze on bacteria and algae. They have two ways of bringing food into their cells, either through special feeding structures such as oral grooves, or they can phagocytize it and bring them in and then lyse that structure or then break that structure down within the cell. When we're talking about a parasitic protozoa, they can live on the fluids of their host. Now, what would be the most limiting factor for these cells, and you think about the way that they live, it's actually they're limiting growth factors availability of moisture. So they generally live in fairly moist environments, such as fresh and marine water, soil, plants, and animals. But regardless of water, if water is readily available, they can live in very high temperatures or very low temperatures and very extreme pHs. And protozoa can also create a spore-like structure known as a cyst, and we're going to talk about that here in just a minute. One last thing to hit on this with this extreme environment that they live in, for example, we talk about Cryptosporidium a lot in our valley. Cryptosporidium can't be killed by chlorine, which is why it spreads so readily through public water parks and fountains and things like that. In fact, they have to have a UV filter. We would have to put so much chlorine in the water that it would actually burn yours and my skin before it would actually kill the Cryptosporidium. So remember, extreme pHs and temperatures, not a big deal, but they do need water. So let's talk about the spore that they form, and these spores are called cysts. Remember, these cysts are just unlike spores, which bacteria formed a special body to create. Cysts are the original cell body, just rolled up into a cyst, and you can see that structure forming here. We start with the trophocyte, and it becomes a cyst-like structure, and which is a dormant resting stage when the conditions are unfavorable. They're resistant to heat and drying, very, very resistant, just like we talked about before. When these cells recognize that the environment is right, they can come back into, they can germinate and come back into the trophocyte again and live like a vegetative cell. Remember, just like how not all bacteria form spores, not all protozoa can form cysts. So not all of them can do that. But these are generally how parasitic protozoa live, and they can be passed from person to person via cysts. So now let's look at the pathogenic. Let's wrap up this topic by looking at the pathogenic protozoa. We have three main types. We have the ciliated, we have the flagellated, and then we have the apocomplexian. These are different types of motility. So check them out. Some of them look pretty familiar. We got Giardia on there, just as we talked about. We got Cryptosporidium on there, just like we talked about. I don't expect you to memorize the list, but I do want you to pick out one or two to remember about each one. Think about the big ideas on each of these. As I said, I'm never going to ask you about a specific organism, but know what the different groupings are. This is the end of this topic. If you have any questions for me, please let me know.